Je pense qu'on commence en fait. En tout cas, j'ai le plaisir de vous présenter Pierre-Marc Delaux. Pierre-Marc Delaux, c'est un chercheur qui travaille à Toulouse. Je pense qu'il a fait sa carrière à Toulouse. Il a fait un postdoc en Angleterre et aussi aux États-Unis. Donc, Pierre-Marc est une personne qui a une trajectoire impressionnante. Il a beaucoup de prix en fait. Il travaille sur les symbioses. Je pense qu'on va écouter pas mal de choses sur les symbioses aujourd'hui. Et donc ici, quelques prix qu'il a obtenus. Et surtout, je connais Pierre-Marc parce qu'il a beaucoup d'articles. Euh, il est pas mal, il a beaucoup de citations, en fait, des articles. Ici, on a quelques Google Scholar, mais aussi, je connais ces articles depuis longtemps qu'on partage, on discute avec nos étudiants. Donc, on voit ici quelques exemples. J'imagine qu'il va parler de quelques articles. Donc, pour moi, je suis très content de qu'il a accepté de nous parler. Et en fait, je te remercie parce que c'est un peu tard en France. Donc, voilà, je te laisse la parole et merci.
Merci beaucoup, Juan Carlos. Donc, je pense que je vais switcher en anglais parce que j'ai l'habitude de faire cette présentation en anglais. Je peux aussi, bien sûr, refaire certaines slides en français si c'est plus approprié. So thanks a lot, Juan Carlos, for the invitation. It's really a great pleasure to be with you this evening for me to talk about our research in Toulouse. So my lab is based in Toulouse, as Juan Carlos said. It's a lab which is funded by the CNRS and the University of Toulouse. And my group there is really interested in plant microbe interaction in general. We are doing a bit on plant immunity, trying to understand how plants evolve a defense mechanism. I won't talk about that today. And the other large topic we are exploring is really symbiosis and the evolution of symbiosis in plants. And this is what I'm going to talk about today. I try to come with a number of different stories, illustrating a bit the type of approaches we run in the lab, which are, as you will see, quite diverse. So just a very brief summary of the plant diversity. I'm sure you are all familiar with that. So here is a summary. So it's not an actual phylogenetic tree. Actually, for instance, the charophytes here are paraphyletic. So it's just a summary. But basically, plants can be divided in two main groups: the green algae, which are mostly aquatic organisms, although there are a few terrestrial algae, and the land plants or embryophytes, which are formed by two main clades, which are monophyletics. One is the laser pointer here. One is the non-vascular plants, which are also called bryophytes. And in the non-vascular plants, I really ask you to remember the liverworts, like Marcantia that you have here. And then the other clade is composed of vascular plants, such as flowering plants, angiosperms, or ferns, for instance. And obviously, these plants are quite amazing for a number of reasons. And the main one being that they are really primary producers of biomass by fixing atmospheric CO2 and by directly mining nutrients in the soil. That makes them quite unique. But obviously, my interest is not really in understanding how plants get these nutrients and so on from the environment. It's more the other way they have evolved to get these nutrients that really fascinates me, which is the symbiotic way of getting nutrients. And in a way, this extremely diverse set of plant organisms is mirrored by a huge diversity of symbiotic associations. And when I say symbiotic associations, I mean really mutualistic association, where the two partners benefit from the association with the other partner. They can be divided, these associations, in two main groups. The intercellular association, and here you have a cartoon of a plant cell, an algal cell. It's a chlorophyte, actually, which is surrounded by a fungus growing outside of the cell. And this is typically the kind of association we will find in lichens, which are found in some chlorophyte green algae. This is a symbiosis we are just starting to study. And then there is another type of association, which is much more intimate, where the partner, for instance, the fungal partner, in the case of the arbuscular mycorrhizal symbiosis, the fungal partner colonizes intracellularly or transcellularly the plant host cell. And it leads to plant cells perfectly alive, hosting within their cell wall an actual microorganism. And this is really an extremely intimate association. Of course, the arbuscular mycorrhizal symbiosis that you have here is not the only intracellular symbiosis. There are other intracellular symbiosis with fungi, and here you have different types, and I will discuss just a little bit at the end of my talk about this association. But there is also intracellular association with bacteria, like in the case of the nitrogen-fixing symbiosis formed in few angiosperms, including legumes, where the rhizobia are hosted inside the plant cell again, where they fix nitrogen and deliver this nitrogen to the plant. So a huge diversity of plants, which is mirrored by a huge diversity of symbiotic types. And I think if you are not fascinated by this session, as I am, I think this picture really will, I hope, will get your attention. So this is really the arbuscule I was discussing just before. It's a picture that was taken a number of years ago by Marc. And in this picture, you really see the intercellular hyphae formed by the fungus that colonizes one plant cell here. And you see the extremely branched structure formed by the fungus inside the plant cell. So again, that's not a pathogen which is eating a plant cell. It's really a symbiotic fungus inside the cell, with the cell actually accommodating this fungus. And what's really magic about this association is that if you take the same cell and come back in a few days, you won't see the arbuscule anymore, because the cell is able to de-differentiate back to a non-symbiotic state. So this is a transient mechanism. So as you may guess, to make something so special, it needs a number of finely regulated mechanisms to allow this association. And we can briefly divide these mechanisms in three main symbiotic steps leading to the arbuscular mycorrhizal symbiosis. So first, the two partners are growing in the soil. So here you have the plant roots, for instance, of a legume, and the fungus is in the soil, and they start exchanging chemical signals. I will briefly discuss about these signals later on during my talk. And these signals, on one side from the plant, activates the fungal metabolism, so it really sort of wakes up the fungus. And in exchange, the fungus produces other compound that activate the plant symbiotic program. Once this program is activated, the fungus contact the plant cells, the epidermal cells on the root in vascular plants, and the plants form these accommodation structures, really allowing the fungus to start invading the epidermis. Later on, the fungus reaches the cortical cells, where it develops these highly branched structures. And again, that's really the plant which is controlling the formation of these intracellular structures. And at these specific sites here, the two partners exchange goods. So the fungus is mining nutrients and water in the soil and delivers it through these arbuscules to the plant. And in exchange, the plant delivers mostly carbons, which are either sugars, but also lipids. And this is important. That was discovered now 4 years ago, that angiosperms deliver a lipid to the fungus. And that's really important. I'll come back to that later. So obviously, these associations, they are extremely important in extant ecosystems, in agronomic ecosystems, but also in natural ecosystems. But they have been even more important in the past, I think, and in particular for one event that we call the terrestrialization event, which is really one of these few key transitions during the history of Earth that changed everything in a way. And this terrestrialization event is the colonization from an aquatic environment, probably a freshwater lake or something else, a pond, let's say, to the terrestrial soils, the the actual lands. And this transition of plants from aquatic to the lands really completely modified the geochemical cycles, pumped oxygen in the air, led to a creation of an actual soil, which was not really present before plants colonized lands. And this event took place here as a base of what we call the land plants today. And that was approximately 450 million years ago. And it had been proposed maybe 50 years ago now that this colonization event actually required the association of plants with symbiotic organisms, symbiotic fungi. That's definitely not the only innovation that plants had to evolve to colonize lands, but that's probably one of these associations. So why do we think this arbuscular mycorrhizal symbiosis was one of these terrestrialization innovations? The first reason is that we find this association, in 80% of the extant land plant species. So basically, almost all land plants have that type of arbuscules in their cells. And in most of these cases, nutrients are delivered from the fungus to the plants. So it's beneficial to the plants in most of these cases. A really stunning result also from the late 1990s is that in the most ancient macrofossils, plant macrofossils, so really the earliest plants that look like a plant that we have access to, in these fossils, in sections of these fossils, were found that type of structures that really look like arbuscules that we find in extant species. The picture I showed you a couple of slides ago. So it really looks like 407 million years ago, at the age of this fossil, there was already that type of structures in plant cells. And altogether, that really suggests that the arbuscular mycorrhizal symbiosis was present in the most recent common ancestor of all land plants and probably played a role during this terrestrialization process. So now in my group, we are really trying to understand how this association evolved that long time ago. And we are also interested in understanding in extant species, what are the mechanisms, the molecular mechanisms that allows this intracellular symbiosis, with the idea that if we understand how it works, we will better understand how it evolved. And actually, we can reverse this thinking that if we better understand how it evolves, we will also better understand how this association works. So our working hypothesis is that we will find essential mechanisms, symbiotic mechanisms, by looking at the conservation of symbiotic mechanisms across land. The first thing we did, and we started a number of years ago when I was still a postdoc, actually, we took the genes that we know are essential for this association, the arbuscular mycorrhizal symbiosis, in angiosperms. So we know pretty much 20 genes, 25 genes, essential in angiosperms for these associations. And we sequenced quite a number of genomes in all these plant clades and looked for these genes in all these genomes, either in collaboration with other groups or genomes that we sequenced ourselves. And here, the first take-home message is that all these genes but one that we knew from angiosperms are also present in bryophytes and in a number of other vascular plants we looked at. So clearly, all these symbiotic genes, they were present in the most recent common ancestor of the embryophytes. There is type here. I want also to say that we looked at a number of streptophyte green algae that I summarized here as charophytes. And we also found a number of these genes in green algae, suggesting that the evolution of this symbiotic gene set here is a mix of recently evolved genes and genes that were already present in the common ancestor of green algae and embryophytes, and that got co-opted for the evolution of symbiosis. I won't talk that much about these genes today. So basically, we had all these genes present here in the most recent common ancestor. However, the fact that these genes are present here, or were present here, because they are conserved in all land plants, doesn't mean that they were actually playing a symbiotic function in this ancestor. And the only way to determine whether there were actually symbiotic genes, and there have been symbiotic genes from the beginning, would be to compare what we know from vascular plants to models in bryophytes, where we could also conduct a genetic analysis. Because if we find that these genes have also a symbiotic function in bryophytes, such as liverworts, we could then infer that their most recent common ancestor was also using these genes for symbiosis. And that's really something we started now 6 years ago. And we really took the time to develop a model here in the liverworts to address that particular question. And the model we picked is called Marcantia palliaceae. So that's how these liverworts look like. It's a leaf-like structure growing on the surface of the soil. And if we make a section of that structure, that's how it looks. It has an upper epidermis here, or dorsal epidermis, where photosynthesis takes place. Then the plant is anchored to the substrate through these rhizoids, which are root hair-like structures. And in between, there is a parenchymatous tissue. And the fungus colonizes the plant through the rhizoids. When it contacts the base of the thallus, it forms intracellular hyphae and ultimately these arbuscules here in the middle of the plant. And there is a very dedicated area where arbuscules are formed. Now, if we look at the sections under a confocal microscope, that's how it looks like, with the rhizoid in cyan, so that's the plant structures, colonized by the fungus, which is stained with a green fluorescent dye. And this is the fungus colonizing the rhizoids, forming this intracellular hyphae, and then reaching this dedicated part of the thallus where it forms these highly branched intracellular structures. So it's really an intracellular symbiosis in Marcantia. So we had this Marcantia, this liverwort that was well colonized. We sequenced the genomes. We spent a lot of time developing genetic transformation, genome editing, and with all these tools, then we started addressing the question whether these genes, these symbiotic genes from angiosperms that were present in the most recent common ancestor, do they play a role, a symbiotic function in Marcantia? So the first thing we did, and that's the work from two quite amazing postdocs from my group, Melanie Rich, who did all the wet lab experiments, and Jean Keller, who is a computational biologist doing all the phylogenetics I talk about. So they took a Marcantia, inoculated it with symbiotic fungus, and compared the transcriptome of these inoculated plants versus non-colonized plants. And they looked at this RNA-seq data over 3 time points, 3 weeks, 5 weeks, and 8 weeks post-inoculation. And obviously, we found more than 1,000 genes that were differentially regulated during this time course inoculation experiment. And here I just picked 3 biological processes that were highly enriched in our RNA-seq data. It includes the carbohydrate metabolism, ammonium transporters, and proton export. So that's typically what we expect from mycorrhizal plants. It will induce the carbohydrate metabolism to feed the fungus, express ammonium transporters to get nitrogen from the fungus, and this proton export mechanism is used to actually energize the ammonium transporters. So it looks like there is a sort of expected transcriptomic response to the fungus in Marcantia. So what we did after that was to map these transcriptomic changes on a phylogenetic by comparing the expression of the orthologs of these Marcantia genes in angiosperms. So we picked 5 different angiosperms for which we had RNA-seq data during mycorrhizae, and we looked whether we find the same genes also upregulated during Marcantia. And here I show you just for these 3 processes, but it was even more obvious with other biological processes. We found that many of the genes upregulated during symbiosis in Marcantia were also upregulated during the symbiosis in the other species, and that's these black squares you see here and there. So it seems that there is a universal transcriptomic response to the arbuscular mycorrhizal symbiosis in plants. Now, one particular process really took our attention. And this is the biosynthesis of fatty acids. And here you have just a few genes, and you see that this upregulation of the fatty acid biosynthesis is really conserved across plant species which are engaging in symbiosis with the arbuscular mycorrhizal fungi. And that's true in Mercantia, that's true also in angiosperms. So there is very likely production of fatty acids in plants when they are colonized by the arbuscular mycorrhizal. What was even more striking was the activation of these genes here, the STAR and RAM2, which are well known in angiosperms, because RAM2 modifies fatty acids into monoacylglycerols, and these compounds are known to be, or supposed to be, transported through STAR and STAR2 to the arbuscular mycorrhizal fungus. So this is the way the plant in angiosperms delivers lipids to the symbiotic fungus. And we found the same pathway activated again in Mercantia during symbiosis, suggesting that this lipid metabolism might be, or the symbiotic lipid metabolism might be something extremely conserved across plant species to feed the fungus. And this is actually, I think, something even more than important. It is something probably essential. Because if we look now at the fungal phylogeny, so here you have a summary of the fungal phylogeny with here the arbuscular mycorrhizal fungi. And when Jean looked at or compared these genomes here, really striking features of this arbuscular mycorrhizal fungi is that they don't have the fatty acid synthase. So they lost it very early before their diversification. And this fatty acid synthase complex, what it does produce fatty acids. So in one way, on one side, we have the fungus that very early during its evolution lost the ability to produce fatty acids. And on the other side, plants that seem to have evolved a dedicated metabolism to produce lipids to feed the fungus. So we think that's really sort of the art of this symbiotic process that maintain the association mutualistic. Of course, this is only based on expression data and comparative phylogenetics. So we needed to go to deep into the mechanisms to really test that hypothesis. And this is what we did. So first, Nicolas, who was a PhD student in the group, looked at the accumulation of lipids, actual lipids, during the association in Mercantia palliaceae. And in particular, he looked at triacylglycerols, which is a class of lipids accumulated by the fungus in flowering plants. So here you have the quantification of a number of these compounds in non-mycorrhized Mercantia. And upon colonization, 8 weeks after colonization, we saw a huge increase in the accumulation of these compounds in the mycorrhized plants. And what was striking is that here using a dye, a Nile red dye that stains lipids, so chalcofluor is used to stain the fungus in cyan and the plant cell wall. And you can see on this picture that the fungus is basically full of lipids. So Nile red indicates lipids, and the fungus is full of lipids, although we know it's not able to produce its own fatty acids. So here it really suggests that the plant is definitely delivering lipids to the fungus as in angiosperms. However, that's not direct proof of that transfer. To actually test this transfer really directly, we used an approach that has been used in the past in angiosperms. So what we did, or what Nicolas did, was to express this gene called UC-FATB that stops the elongation of lipids at the C12 stage. Usually plants produce C16, and using this enzyme, we can modify the fatty acids they produce and enrich in C12. And if there is an actual transfer of lipids from the plant to the fungus, we should see an enrichment also in C12 in the fungus itself. And to make the system more specific, we drove the expression of this enzyme using a promoter which is specific to the arbuscules. So here you have a section of Mercantia. This is the area where the arbuscules are formed, and you can see that promoter Gus fusion is really dedicated to these cells. So we have a system which is only active in the arbuscules. So Nicolas grew Mercantia, colonized with fungus, and separated the two partners by a membrane, nylon mesh. And that way we can separate the colonized plants and the fungus. So Nicolas looked at the lipid composition in the colonized Mercantia, and he found that the engineered lines that express this UC fatty gene accumulated trialkylglycerols with 3C12. So they are, and this is not something we detect in the wild type. So that means that our engineering trick works in Mercantia. He also looked at a very specific fatty acid or triacylglycerol that we know is specific to the fungus. It has a 2 in saturation on C16s and 1 C12. And we found this compound only in, again, the engineered lines. So showing that the fungus does take up lipids in the plant and that we can detect it using our C12 modification. Now, if there is indeed transfer of lipids from the plant to the fungus, we should be able to detect the same type of lipids in the fungus only compartment. So Nicolas collected the fungus here and trust me, to get enough material to do lipidomics on the fungus, it takes quite a number of pots. So he collected this material and extracted the lipids and looked for the different triacylglycerols. And what he found is a very striking presence of this compound in the engineered lines, really demonstrating that the fungus is directly taking up the lipids from the plant. Now, we know that the mechanism is present in Mercantia. It doesn't mean that it's the same than in other land plants. So we need to go down again to really the molecular mechanisms. So Melanie went back to the RNA-seq data and looked for transient factors that could be involved in the activation of this lipid metabolism. So she compared the data in Mercantia to the data in angiosperms, and she found 2 transcription factors, one here and another one here, that were upregulated in all the species we looked at. And this one here is called Mercantia wrinkled. So we also studied that one, but so far we didn't see, or at the time we didn't see any. So we picked up Mercantia wrinkle and tried to test whether it might be linked to the transfer of lipids. So first, Melanie generated promoter Gus fusions using this Mercantia wrinkled and looked at non-mycorrhized Mercantia. And you can see that in this plant, the promoter is active in the upper epidermis. And then when the plants were inoculated, in addition to this staining on the upper epidermis, there was a clear staining that you can see here in the area where the arbuscules are formed. So the wrinkle is expressed during mycorrhizae in the area where arbuscules are present. And that was also true for RAM2, which is this gene just downstream or involved in the lipid metabolism. Next, Melanie overexpressed wrinkles in Mercantia and generated RNA data for these overexpressing lines in absence of the fungus. And here, that's just a reminder that the lipid metabolism is upregulated in Mercantia during mycorrhizae, so that's these 3 first columns. And here in these 4th columns, you have the overexpression Mercantia wrinkle in absence of the fungus. And you can see that there is quite a number of genes which are upregulated in absence of the fungus. And that's also true for STAR2 and RAM2C genes here, showing that we can sort of mimic part of the symbiotic response just by overexpressing wrinkled in Mercantia. And using a number of experiments, we demonstrated that this activation of the lipid metabolism by wrinkle is through the direct binding of wrinkle to the promoter of this lipid-related genes. So now, obviously, if wrinkled does regulate this lipid metabolism, we have a tool to test whether this lipid metabolism is actually essential for mycorrhizae in Mercantia. And that tool would be to just knock out wrinkle and to look at the symbiotic phenotypes of these plants. So here you have a section of Mercantia wild type with the chloroplast autofluorescence, so the upper part of the thallus, and stained in green is the fungus. And you can see that in a section of the wild type, there is plenty of arbuscules which are formed. And if you look under the transmission electron microscope, you can see all these fine branches which are basically arbuscule branches. If we count the number of thalli of plants that hosts that type of arbuscules, basically all the plants, all the wild-type plants were harboring that type of arbuscules. By contrast, when Melanie looked at 5 independent CRISPR-Cas9 knockouts of wrinkle, she did see a little bit of fungus. So the fungus is inside the plant and it's intracellular, you can see it here, but she didn't see a single arbuscule, really demonstrating that wrinkles that regulate the symbiotic lipid metabolism is essential for a functional mycorrhizae in Mercantia palliaceae. And here I have 2 lines which are just small in-frame deletions, and they don't show major phenotypes, really demonstrating that it's the knockouts which leads to these phenotypes and not just the overexpression of the Cas9. And then to sort of finalize this story, Jean looked at the evolution of this transcription factor, wrinkle, because we know the lipid metabolism in itself is something extremely common in green plants, including algae. And what Jean found is that wrinkle is actually absolutely specific to land plants. So here is a phylogeny, and I won't go into the details, but basically here in this large wrinkle clade, you don't see any blue species. So you have only a green species, and blue are either streptophyte algae or chlorophyte algae. And basically what it means is that the evolution of the wrinkled clade is really an innovation of the land plants. And that what we think is that evolution of this transcription factor, specifically in land plants, allowed the recruitment of the existing lipid metabolism to feed the fungus. And that was one of the essential key to evolve this symbiotic association with fungi. So I just discussed about this lipid metabolism, which is one of the 3 steps important for the arbuscular mycorrhizal symbiosis. We also looked at the other 2 steps. So I'll now present our data, which are unpublished, on the role of signaling compounds, strigolactans, in these associations. So in angiosperms, strigolactans are produced via one enzyme called CCD8, and this strigolactan really activates the fungal symbiont while the symbiont is in the soil. So one experiment we did was to compare first Mercantia species that do mycorrhize, like Mercantia palliaceae, Imaginata, or Piñata. So you can see that they have arbuscules, and compare them to Mercantia polymorpha that do not form mycorrhize. And in these different species, we looked for CCD8, this enzyme essential for strigolactan biosynthesis, but also for the actual presence of strigolactan by mass spectrometry in collaboration with the Nomura's lab in Japan. And to make it short, what we found is that CCD8 and strigolactans are specifically present in the mycorrhizal Mercantia species and not present in the 3 Mercantia polymorpha subspecies that do not mycorrhize. So there is a link between the presence of this compound and the genes and the ability to form mycorrhize. Then we took Mercantia palliaceae, and here you have the wild type, which is well colonized, and we generated CRISPR mutants for CCD8. And in these mutants that do not produce strigolactans, we don't see any arbuscules anymore, any infection anymore, actually, as you will see on the next slide. But when we applied synthetic strigolactans on these plants, we were able to fully rescue the phenotype. So really, the presence of the molecule strigolactans is essential for the arbuscular mycorrhizal symbiosis in Mercantia as it is in angiosperms. And to understand at which step of the symbiotic process the strigolactans were playing a role in Mercantia, we quantified the infection points. So infection points are these rhizoids which are infected by the fungus and forms intracellular hyphae at the base of the thallus. So we quantified the number of events per thalli, per plants, in the wild-type plants, or in single and the double CCD8 mutants. There are 2 paralogs in Mercantia. And what we clearly found is that we didn't see any infection points in Mercantia, indicating that strigolactans are essential to make this initial, very initial infection events. And when we applied exogenous trigolactones to these plants, we were able to rescue the phenotypes, really confirming this pre-symbiotic function of strigolactone in Mercantia. So like for the transfer of lipids, it seems that this symbiotic, this chemical dialogue, which is essential in angiosperms to form the association, is also essential in liverworts, suggesting that their most recent common ancestor here again was using strigolactones as a chemical symbiotic signal. Obviously, I discussed about this last step, about the first step, but we also looked at this intermediate stage here, which is all these signaling events necessary to form this intracellular infection structure. And in angiosperms, these genes have been very well described, and they form a signaling pathway. And it's composed by receptors at the plasma membrane, and a number of other components, including a kinase and a transient factor. And what we know is that when the plant perceives the fungus, perceives chemicals produced by the fungus through these receptors, it activates the phosphorylation of cyclops, the transient factor, by this kinase. And this phosphorylation activates the transient factor, which is able to bind to cyclops responsive elements and to activate the expression of the genes which are regulated by these elements. And these genes are basically many symbiotic genes. So that's a way to connect the perception of the fungus to the activation of the plant symbiotic program. And we have a biochemical trick, I would say, to bypass the perception of the fungus. To do that, what we do is to make a point mutation by classical cloning methods in cyclops. And these point mutations, there are two of them, mimic the phosphorylation of cyclops. And when we overexpress this cyclops with a point mutation, it auto-activates all the genes that are regulated by this cyclops responsive element. And if we fuse this cyclops element to a Gus reporter, that's how it looks like in the Medicago roots. We have activation of the reporter, and the roots turn blue. So that's a very easy way to test the function of cyclops. And we can do that with cyclops coming from different species to determine whether this key regulator is really conserved across plants. And Tatiana did this essay cloning cyclops from a number of species. And she used a number of angiosperms, but also liverworts and even carothers. So for angiosperms, all the cyclops tested, and here I show you 5 of them, do activate the Gus reporter. So it seems that in angiosperms, cyclops is extremely conserved at the biochemical level. She also tested Mercantia cyclops, and again, she found the activation of the reporter, indicating that in all land plants, this gene has a conserved biochemical activity. And it's not on the slide, but I can tell you that cyclops from a number of different streptophyte algae does the same. Then Tatiana turned to Mercantia itself and tested the same activation assay. In the line transformed just with the reporter, she didn't see any activation. But when she overexpressed cyclops from Mercantia, again, she got the activation of the reporter, showing that whatever the plant species we take and the cyclops we take, it always activates this reporter. So it's extremely conserved at the biochemical level. Next, using this reporter, Tatiana looked for the activation of cyclops in plants. In non-inoculated plants, she didn't see any activation. So you see a blue dot here, and that's the only section where we saw staining in non-colonized plants. But upon inoculation with the symbiotic fungus, we do see activation of the cyclops reporter, indicating that this symbiotic signaling pathway is activated where the fungus colonizes the plant. And then to finally test the role of cyclops during symbiosis, she made a cyclops mutants. So here you have a section of Mercantia wild type stained with the fungus stained in blue, and you see plenty of arbuscules. But now in any of the 22 alleles Tatiana generated, we don't see well-formed mycorrhizae. In most cases, the plants are not colonized at all. And when they are colonized, like in this one, there are a few intracellular hyphae, a few arbuscules, so there are arbuscules, but these colonization events don't spread in the plant like they do in the wild type. Cyclops, as in angiosperms, is really important and required for a functional arbuscular mycorrhizal symbiosis in Mercantia pallidis. So altogether, that shows that all the key steps to form the arbuscular mycorrhizal symbiosis are present in bryophytes, and thus, because they are also present in angiosperms, were present in their most recent common ancestors that lived on land more than 450 million years ago. So that's really the first demonstration that mycorrhizae is indeed ancestral, because we can track DNA. We can't really track, morphological features, but DNA, we can track it, and we can track its evolution. So that's really the demonstration that mycorrhizae is extremely ancient. And I think one thing to realize here is that if you look at fossils, we don't really have fossils which are, from this time point here, back more than 400 million years ago. We have, at best, we have a few spore tetrads and dyads from a few fossil sites. So we have no idea how these plants look like. But what we know for sure is that they had all these genes I just mentioned, and that these genes were involved in forming this association with symbiotic fungi. And in a way, I found that really amazing to think that we don't know how the plants looked, but we don't know that they were forming symbiosis and which molecular mechanisms they were using. So we have done all this work to really understand the origin of symbiosis, but we tried also to expand this work beyond just liverworts and angiosperms, and to better understand symbiosis in general. So I'll go quickly through this last slides, which is a work done only using a comparative phylogenetics. So here, and it's a quite old paper now, we looked at the genomes of species that form mycorrhizae and compared them to genomes that do not form mycorrhizae. And originally, I did that in angiosperms. And what we found at the time is that species that have lost mycorrhizae in angiosperms, they have also lost most of the genes I presented earlier during my talk. So these symbiotic genes are lost in species that lost mycorrhizae. And this is due to a process that we call co-elimination. So really briefly, co-elimination is a process which is involved here. And basically, if we look at a genome at a given time point, it has this given gene content. Now, during the evolution of this lineage, a trait is lost, whatever it is, symbiosis, for instance. If we come back a few million generations later, many of the genes will have been conserved because they have multiple roles and they are still under a purifying selection. But now there are genes that have been lost. And these genes have been lost because their only function was to regulate this trait that has been lost. And this is a co-elimination where selection on these genes is relaxed because of the loss of the trait. And this is what we call this co-elimination principle that has been also used, for instance, when studying cetaceae that have lost hair, and they have also lost the genes involved in hair development. So that's something, a process, which is, maybe you could turn off your microphone. There is a microphone which is on. Anyway, so this is the co-elimination process that links the loss of a trait and the loss of the genes that are involved in this trait. Okay, so we have tracked these genes present absence that I will represent with the following schemes. So this red cross means the gene is absent, a green square means the gene is present, and when there is a white square, it means we didn't detect the gene, but we used only transcriptome data, so we cannot conclude about the presence or absence of the gene. And we use this comparative phylogenetics method to track all these genes in different plant lineages. So I won't show you all the data. We have done that really in a number of genomes, in angiosperms, of course, but also in monilophytes, for instance, and also in liverworts. And in liverworts, you remember I discussed about Mercantia polymorpha that has lost mycorrhizae, and we found that all these genes that I have shown here and many others are all lost in Mercantia polymorpha that has lost mycorrhizae. So it looks like this correlation we had in angiosperms, loss of the mycorrhizae, loss of the symbiotic genes, holds true also in Mercantia and in basically all land plants. Now we kept looking for more and more genomes, trying to finalize this study, and we actually had already written most of the manuscript, and we started to find these results that sometimes you don't like to find. They are the results that tell you your hypothesis is just totally wrong, completely wrong. And basically what we found is that there are a number of species that have lost the arbuscular mycorrhizal symbiosis, like lupinus, like these orchids here, or this Junger manialis here. They have lost a number of the symbiotic genes here, but there are also other genes, like cyclops, that I presented, which are absolutely conserved in this species. Also, they don't form the arbuscular mycorrhizal symbiosis. So we have really a list of species where our beautiful correlation, well, was not working anymore. And we had really a hard time first discovering that we were wrong, but that's science, but then trying to understand why we were wrong. And actually it took us a few days, but we realized what was going on. And basically, there is something that unifies all these species. And this thing is that all of them, no exception, they have lost the arbuscular mycorrhizal symbiosis, but they have also transitioned to a different intracellular symbiosis. And here, for instance, lupinus is able to form the nitrogen-fixing symbiosis with rhizobia. Orchids are able to form the orchid symbiosis. Acrometrium form an association with Micromycotina. Junger manialis form an ericoid-like symbiosis, and so on and so forth. And while, for instance, Mercantia polymorpha didn't evolve any alternative intracellular symbiosis, so all the genes are gone. And what it tells us basically is that these genes that are present only in species that form intracellular symbiosis are very likely forming a core pathway, a core signaling pathway that regulates in all land plants all the intracellular symbiosis, which are so amazing when the fungus or the bacteria is living, is invading the plant cells intracellular. So we really think that we have found a universal pathway for intracellular symbiosis in plants, and that on top of this universal pathway, alternative or different mechanisms evolved to lead to these different types of association. And this is really this diversification of association that we are now trying to understand as part of this ERC-funded project. So that was my last slide. I'll just finish with a few acknowledgements. So the data presented are mostly the work of Melanie Rich for the lipid and the strigolactone story. Tatiana did the work on cyclops in Mercantia. Jean is our, let's say, guru in a way for comparative phylogenomics, and he did all the phylogenies and the phylogenetic works. I want also to thank Cyril Libourel, who did all the RNA data. And Nicolas Vigneron did the biochemistry for the lipid projects. And of course, all that was possible thanks to a number of collaborations I just show here. And obviously with fundings, and including quite generous fundings from the ERC, the ANR, and the ENSA project, which is funded by the Bill and Melinda Gates Foundation. And I just finished by saying that in this particular project, we are trying to just gather all this knowledge we are getting on the evolution of symbiosis to engineer novel types of associations that would benefit and potentially completely change agriculture in a more sustainable way. And with that, I would thank you for your attention. I've been maybe a bit long, and I'm really happy to take any questions you may have. Thank you.
Merci beaucoup, Pierre-Marc. C'est une conférence extraordinaire.